students, today I welcome you to this virtual science lab. So, in our theory classes, we have learned that these are some of the major components of food, that is carbohydrate, protein and fat. Now, they are very important for our body. We know that carbohydrate and fats are energy giving food. So, they give energy to your body. Then, protein is a bodybuilding food. It helps to build your body. So, there are various components which could be tested in the food. For testing any food item, we need to have a sample. So, here we are taking sample for protein as pulses. For testing fat, the sample we are taking is butter. And for testing carbohydrate, we are taking a sample bread. We know that bread is rich in carbohydrate. We know that butter is rich in fat. And we know that pulses is rich in protein. So, we will start the test. Before we start any test, children, we should prepare the solutions required for the test. And also, we have to prepare the sample itself. Let us talk about the test for protein. For this, we need to prepare two solutions. First, that is solution of copper sulfate, and second, solution of caustic soda. Now, copper sulfate actually comes in crystal form. It is a blue color crystal. So, it is dissolved in water to form copper sulfate solution. Now, caustic soda comes in pellet form, and 10 gram of caustic soda is dissolved in 100 ml of water to form a solution which is ready for testing protein, that is caustic soda. For testing this protein, we need a sample, that is pulses. And whenever for testing protein we are taking a solid sample like pulses I am taking, we need to grind it and make it a fine paste of it before adding this solution to it. So the first step is to add the sample in mortar and pestle and grind it. Grinding means we are making a fine powder of this sample. So once the grinding is done, what we need to do, we need to take a test tube. And now this test tube is filled with water. Now this sample is added to it. After this sample is added in water, then we need to add first sample, that is copper sulfate, and the amount that we need to add is only 2 to 3 drops. So we will take copper sulfate and add 2 to 3 drops into the test tube. Then we will take caustic soda solution and we will add 10 drops of caustic soda to the solution. Now wait for some time, and then we will see gradually the sample which was colorless due to water gradually changes its color and changes into violet coloration. Now violet coloration indicate that there is a presence of protein in the sample. So this is an experiment which proves that protein is present in the food sample. So now moving on to the next component, that is fat. So how to test fat in a food? It is a very simple test. This test you can perform at home also. For this test only we need the sample and a piece of paper. So if we take a piece of paper and then here sample I have taken butter. So if I put some butter into this paper, the portion where I rub this butter will gradually appear transparent as compared to the rest of the paper. So we can easily see through it. Transparent means it is easily seen. So we can easily see. If I put this, now we cannot see through this, but when I put it near this, it becomes translucent and we can see through this. So this is a test. Fat sample can make a paper translucent. So this is a very simple test you can also perform. So if you want to test whether your nuts are having fat or not, you can just take your nut and break it and then just take a piece of paper and crush it into it like this and try to crush it little harder. So if it is having a fat, the fat will come out from the nuts and then you will see the impression of fat. Yes, you can easily see the impression of fat. So this is a simple test which which will prove that fat is present in the sample. Now, third is how to test carbohydrate. Carbohydrate, we will take a sample rich in carbohydrate. For example, I have taken bread slices. So, bread, as we know, they are rich in carbohydrate, and for that we need iodine solution. Now, this is a tincture iodine, children, that we use it for cuts and all. So, we have to prepare a dilute solution first. I have already prepared a dilute solution. So, how to prepare a dilute solution? You add tincture iodine in water and then you dissolve that tincture iodine. Now, color of iodine, as you see, it is brownish in color. So, I will just add a little iodine and I will show you the color of iodine. So, the color of iodine, as you can see, this is the color of iodine, that is little brownish. However, if I pour iodine in a carbohydrate sample, like I have placed it in this bread sample, what you see? You see gradually the color of this is changing. This is changing to blue-black coloration. So, this is a common test for carbohydrates. That is the test of starch. This is called iodine test for starch, where iodine, when combined with carbohydrate, it produces a blue-black coloration. So, this is a common test for carbohydrate. So, how simple it is, children, to test these. So, believing without testing is so difficult. So, now since we have tested here, it is very easy for us to believe that yes, the component of protein show purple coloration with copper sulfate and caustic soda, and that fat, when it is rubbed against a piece of paper, it causes the paper to become translucent. And when iodine is added to carbohydrate, what we see? This coloration change, that is from normal color of iodine to this bluish black color. So, this indicates the presence of starch, that is carbohydrate. So, this indicates the presence of starch. So, thank you all of you, children. This is all from today's virtual lab of science. Thank you and have a nice day.